Our next speaker is Priya Rayagopalan from, I don't know how I did on that, but from the Loney at UCLA. And the talk is entitled Carriers of TREM2, Alzheimer's Risk Genes Show Accelerated Temporal Lobe Atrophy and Cognitive Decline.
Hello. Yes, so as you may have guessed, I'm not actually Priya Rajagopalan. I'm her colleague, Derrek Hibar. She's actually just recently had a baby boy, so she can't be here today. So hopefully I can be here in her stead. So the title of the talk is Carriers of TREM2 Alzheimer's Risk Gene Show Accelerated Temporal Lobe Atrophy and Cognitive Decline. And so let's talk a little bit about the TREM2 gene. It actually encodes a protein that's part of a signaling pathway that works with the, sorry, let me see if this works. Yeah. Sorry about that. Yeah, so if these are the functional relationships with genes in the same pathway, and TREM2 is encoding a receptor that works along with the TYRO-BP gene that's involved with inflammation and the clearance of cell tissue and apoptosis. The disruption of these two genes has been associated with some clinical disorders, some mostly rare. There's the Plossl disorder, which has typical onset of dementia and things like that, although the link between Alzheimer's is not exactly clear. Some other things, it's also interesting that it's expressed in microglial cells as opposed to in actual brain tissue. And so all of this renewed interest in TREM2 was sparked by a very recent paper published in New England Journal of Medicine by the Johnson Group in Iceland. They actually studied a huge population of basically everyone on the island. And they identified a variant, RS7593268, in the TREM2 gene. It's, it's relatively rare, actually, so only about 0.3 to 0.4% of the population have this specific variant. And in our study, because that variant is so rare, it's actually hard to determine the genotype at that specific place. So we used a proxy SNP, which is easier to identify and is also highly correlated with the original SNP. So one of the interesting things that came out of the Johnson study was actually that this TREM2 variant increases your lifetime risk of Alzheimer's disease by 3 to 5 times, which is a huge, huge impact. And it's on the same order as some of the other Alzheimer's disease genes like ApoE, which most of us know about and are familiar with. But one of the things that we're interested as human brain mappers is actually how those effects can be visualized and across the full brain. They actually, in the first analysis in the Johnson paper, it was strictly a family-based association study. So what is our hypothesis? We wanted to look at atrophy rates in subjects scanned at baseline and then over time longitudinally. And we wanted to see, we hypothesized that carrying one of these rare variants that increases your risk for Alzheimer's disease will be associated with some of the typical patterns of effects that we see in neuroimaging phenotypes. So that's increased hippocampal atrophy, increased temporal lobe atrophy, and also poorer cognitive performance. So we're using the ADNI study population. I'm sure most of you are already familiar with it, but if not, everything's freely available and it's really great. It's an ongoing study too, so they have thousands of data subjects, and including longitudinal data. For this study specifically, we looked at 666, that's an unfortunate number, but that's how many subjects we had with both time 1 and follow-up scans at 1 year and 2 years that also had genetic data and that were Caucasian. We studied the Caucasian sample because it causes problems to have multiple ethnicities in a genetic analysis. The breakdown of the diagnoses are 150 with probable AD, 327 with mild cognitive impairment, and 190 age-appropriate healthy elderly controls. And for the genetics, they were all of these subjects were genotyped, like I mentioned, and we did the standard filtering criteria. And we then imputed these data to the 1,000 genomes CEU reference set, and that's what gave us this new variant that's related to the original one in the Johnson paper. And we checked all of the other filtering criteria as well in terms of Hardy-Weinberg equilibrium and things like that, and that was all good. The specific phenotypes that we're studying, I wanted to talk a little bit more about tensor-based morphometry, which I'll do in the next slide. So we're actually going to be looking at the temporal lobe specifically instead of the full brain because it's implicated as a phenotype or a biomarker for Alzheimer's disease. So just so we're all on the same page, and I don't know if this is overkill, but TBM is very similar to VBM in some senses. This is a longitudinal analysis, so each subject will have two scans, a time 1 and a time 2, and all of the scans, regardless of time point, are registered into a common template space. And then once they're all in the same space, the differences between subjects time point 1 and time point 2 are subtracted so that you get this 3D map of atrophy. And in general, we can plot these values, these Jacobian values that are actually the distance that's required for that represent the atrophy. And just using a simple color heat map, the voxels that are colored in the warmer colors represent a decrease in atrophy relative to the brain template, and then also the blue, the cooler colors represent a volume excess or a contraction, expansion. So we did a very simple statistical analysis, and it's your classical multiple linear regression, even though it's written kind of strangely. Like I said before, the dependent variables that we wanted to look at in our hypothesis was hippocampal volume, both left and right, temporal lobe atrophy at each voxel. So we're prioritizing only the regions in the temporal lobe, but we're still looking at each voxel in the temporal lobe. And then also some cognitive scores like MMSE, CD total, and what's the other one? ADAS cog. And then the predictor of interest is the TREM2 variant itself, and whether you have it or not. And in this case, generally you have 2 copies of an allele, but because it's more rare, we only have cases where subjects have 1 copy of the variant. So it's basically a binary predictor. You have it or you don't. And then the confounders that we regressed out are H and sex. And of course, we're doing lots of tests, so we're using the standard false discovery rate correction at 5% FDR. Okay, and so here are some of our results tabulated, and you can see across the board there was significance. We're doing permutation-based tests here, so and then the p-values are from the parametric multiple linear regression, and you see in general. The trend, the direction of the effects also makes sense. So, for example, MMSE, for every containing this TREM2 risk variant, means that in 1 year, your MMSE value will go down by 3.57 points on average. And a similar interpretation for the CD total and ADAS cog. These values are positive because the interpretation is inverse from MMSE. higher score in MMSCD is good, a higher score on CD total and ADAS is worse. Similarly for the hippocampal volume, if you have this TREM2 variant, in 1 year you'll have an additional 392 millimeters cubed atrophy compared to those that don't. And for the TBM map, inside the temporal lobe atrophy, inside the temporal lobe voxel-wise, we noticed a trend of 1.4 to 3.3% greater brain atrophy on average, and that's at a FDR critical p-value of 0.004. And so this is actually just an uncorrected map of the voxel-wise beta values from this voxel-wise analysis of the temporal lobe. And you can see in general that there's a wide range of warm colors which represent tissue atrophy, and especially in some of these areas you see very, very significant effects. And like I said before, we're seeing on average in the corrected maps, voxels that stay significant are between 1.4 and 3% greater atrophy per year if you have this TREM2 allele. So what are our conclusions? This is the first neuroimaging study to reveal a detailed 3D pattern of brain atrophy on MRI associated with carrying the TREM2 gene variant. And selection of these carriers can be used to help boost power in clinical trials because their brains are declining much faster. And also, this is interestingly the first imaging genetics study confirming the association of a rare variant identified from genome sequence data. And also, thanks to my boss, and then also Priya, who's right here. And that's it. Thank you.
I'm really totally naive about genetic association studies, but statistically, can you help me explain? You have a 3% risk at the population level if you have this genetic variant, right? At the population, you said there is an increased risk of Alzheimer's disease at the 3% in the general population.
In the general, so that's 3% atrophy, more atrophy per year.
No, but in general, you said that the risk of the TREM2 is similar to ApoE, which is...
Oh, yes. The 3 to 5 times greater risk.
Yeah, 3 to 5, right. Then you said that the particular SNP that you are evaluating is not exactly the one that was studied.
Right.
And there's an association in our score about 50%.
Yeah.
So 50% of the variants kind of get dropped by selecting the SNP. And then you said that you kind of dichotomize your analysis so that you had only basically either you had it or you not had it, irrespective of whether you're homozygote or heterozygote, I guess. So okay, so presumably the risk then decreases even further. I mean, the chance, right? Because now you are, washing out that particular genetic association risk by dichotomizing your population.
Sure.
And then the question is, and then I think, of course, everybody's going to get worse over time, right? Everybody, both the control sample and your experimental sample. So I think what you should be doing is some kind of an interaction between the atrophy scores between the normal decline in the control sample and the further decline in the experimental sample, and only that should be potentially explained by this particular genetic criteria that you selected. And then the question is how that may propagate to other genes. So I guess the general question is specificity maybe and sensitivity of this finding in relation to this different adjustment that you had to do in order to carry out your study.
Sorry, what specific adjustment though? I think it's not unheard of. It's actually a different type of model, a genetic association model, where you group homozygous and heterozygous together. the dominance model. Is that what you mean? I mean, I think the presence or absence of this particular gene variant. I mean, it's in a few people. It's relatively rare. Yeah, I'm not sure how that's.
Your anatomical analysis was an interaction.
It actually, it was, I think, an
was.
interaction analysis. I mean, because he was comparing the percent change in the carriers and the non-carriers over that year, so it's effectively an interaction.
Sure.
Okay. Sorry, in the interest of time, we'll have to move on, but thanks for a great talk.